Voltage gate ions channel 3, channel diversity. Today we're going to discuss the molecular structures and the diversity of potassium channels, calcium channels, and sodium channels. Potassium channels is the most diverse group of channels. Some are voltage dependent, some are calcium dependent, some are hyperpolarization activated, and some are what we call leak channels. The voltage dependent calcium channels usually they fall in the KV channel family. The calcium dependent channels are usually called either BK for big potassium channel or SK channel or small k channels. The hyperpolarization activated potassium channels belong usually to the KR channel family. Finally, the leak channels are also very diverse and they have different names as task track channels. Let's start our discussion with the KV channels. So the KV channels usually have this kind of structure: 6 transmembrane domains, S1 to S6, and a P-loop that forms the selectivity filter of these channels. The polypeptide that encodes S1 to S6 is called the alpha subunit of these channels. Sometimes you also encounter a beta subunit that associate with the alpha subunit, and these beta subunits may modify the gating of these channels. The 6 transmembrane 1-pore type of channels are tetramers. So 4 alpha subunits come together to form a functional potassium channel. And members of this family are KV channels, and also some other channels like the ATERG channels, some channels, the BK and SK channels as well. The 2 transmembrane 1-pore channels are the KR channels. You can see from this diagram that this type of channels having only 2 transmembrane domain in a P-loop, so they don't have the S1, S2, S3, S4 domains, and they are also tetramers. So 4 subunits come together to form a functional channel. Finally, the leak channels, or 4 transmembrane 2-pore channels, have different names such as tweak, track, task, or track channels. And what you can see from this figure is that one polypeptide contains 2 P-loops. That's why it's called 2-pore domain channels. And they usually come together as dimers of these polypeptides to form one functional channel. So what we call transient potassium channels, also called A currents or KA currents, are encoded by KV channels such as KV 1.4, KV 4.1, or KV 4.2. They are characterized by a very fast activation kinetics. It takes just very few milliseconds to activate these channels, and they have a threshold for activation near minus 40 millivolts. Also, these channels inactivate relatively fast. Not as fast as the sodium channels, but nevertheless, it just takes few milliseconds for those channels to inactivate. So these channels, because have a threshold around minus 40 millivolts for activation, they are immediately active at resting potentials. So what's the function of these transient potassium channels? Well, when these channels are activated, no spiking of the cells will occur until these channels are inactivated. And therefore, these channels will dynamically modulate the membrane excitability of neurons. Many of these transient potassium channels are modulated by protein kinases or phosphatases, and therefore by modulating the activity of these transient potassium channels, one neuron may have different excitability patterns according to the status of the cell. The delayed rectified potassium channels are also mostly encoded by other subunits of Kv channels. As you saw in the beginning part of this course, these channels have a more delayed activation kinetics, have a very slow inactivation, and the most important role of these delayed rectified potassium channels is that they allow repolarization of the action potentials. As other Kv channels, they are formed by tetramer of alpha subunits plus beta subunits. Many of these channels are blocked by these many types of potassium channel blockers. Overall, over 40 Kv channel alpha subunits have been described in the mammalian genome. So there is a large diversity in terms of Kv channel heterogeneity in the mammalian neurons. Now let's discuss the two transmembrane one-pore type of channels. Those are the KR channels. If you look in this figure comparing the structure of KR and a KV channel, you can see that the KR channel is much more simplified. It doesn't have the S1 to S4 domains, and therefore they don't have a bona fide voltage sensor. Actually, these KR channels, they look more like the KCSA channels that we talked in our previous lecture. These channels are called KR channels, that stands for inward rectifying potassium channels. The major feature of these channels is that they are inward rectifying. So if you look, the current voltage relationship for these channels, it looks something like this. And the current is equal to 0 at approximately EK. So these channels allow large inward currents to the cell, but very small outward currents from the cell, and therefore they're called inward rectifying potassium channels. They are also formed of 4 subunits coming together to form a pore. And again, one of the major properties of these channels is they are less conducting at positive membrane potentials. Sometimes these channels are referred as anomalous rectified potassium channels because the conductance of these channels increases with hyperpolarization and decreases with depolarization, which is the opposite of what has been described for KV channels. And therefore these channels allow potassium to flow into the cells, and also they allow some potassium to flow out of the cells when the cells are more depolarized than EK. KR channels, also called IRK channels, are very important in regulating cellular excitability, insulin secretion, potassium homeostasis, setting the vascular tone and regulation of the heart rate. The first two members of this large family was cloned in 1993, and they were renamed as KR channels. Why these channels would be important in neurons is because if these channels are expressed, the, the, the main potential of these neurons tend to be clamped around EK, minus 80, minus 70, or minus 60 millivolts. Now, when the cells depolarize by any other reason, for example, by excitatory inputs or so on, now these channels become less important and therefore will not clamp the main potential close to EK. One knowledge that you can have for these channels is if you think about a latch in a door, these channels would serve as a latch to keep the main potential close to EK. Now, when the cell depolarize, the latch release, now the main potential of these cells can move freely to more depolarized potentials. So the bottom line is that KR channels have a very important role in regulating the membrane excitability of neurons. So what accounts for the voltage dependence of these channels? As we said in our previous slide, these channels are inward rectifying. That means that they allow less current in the outward direction than inward direction. If you see the recordings from this cell that express these KR channels, you can see that in the on cell or cell attached configuration, there is this very strong inward rectification of these channels. Now, when a patch of membrane is excised from the cell, so in an inside-out patch configuration, now these channels exhibit a linear behavior. So the inward rectification is conferred by some form of intracellular factor, in this case magnesium, because when magnesium in the intracellular side is restored to 1.2 millimolar, which mimics the intracellular levels of magnesium, the inward rectification is restored. Therefore, magnesium blocks this channels from the intracellular side and confers the inward rectification profile of these channels. More recently, we also discovered that not only magnesium is an important intracellular blocking particle of these channels, but also polyamines. Polyamines such as spermine or spermidine are present in the intracellular environment of these cells at micromolar concentrations. And if you see in panel A, an inside-out patch in the presence of 1 millimolar magnesium, here, the inward rectification is not completely restored by magnesium, because in cell attached, the amount of rectification is even higher. And what is shown in this bottom part of the panel A is that when 10 micromolar spermine is applied to this patch of membrane, now the inward rectification, strong inward rectification profile is restored. And finally in B is shown here the inward rectification currents that's very similar if you apply 10 micromolar spermine or 1 millimolar magnesium on these patches of. So for exercise 1, explain the effects of intracellular magnesium on KR channels. What's the most likely site for magnesium binding for KR channels? If the binding site for magnesium is on the external part of the pore, then what will be the expected IV relationships? These KR channels subfamily contains several members that are listed as KR1, KR2, 3, 4, 5, 6, and 7. The KR1, 7, and 4, they're mostly involved in potassium transport. So many of these channels are expressed in epithelial cells and also expressed in glial cells. And in those type of cells, potassium transport is an important function of these cells. KR2 channels and also KR5, in particular KR2 channels, are the classical KR channels. So those classical KR channels are KR channels that they're usually open at rest main potentials and have that important role in clamping the main potential close to EK. KR3 channels are called G protein gated potassium channels because usually these channels are closed under resting conditions, but when a neurotransmitter binds a G protein couple receptor, activation of G proteins may activate or promote the opening of these channels. KR6 channels form the ATP sensitive potassium channels. Usually these channels are closed at normal resting main potential, but sometimes when the cell becomes anoxic, the intracellular levels of ATP decreases and then there's opening of these potassium channels. And therefore we think that the function of these channels is to provide a neuroprotection to these cells. Here's a diagram showing a GPCR or G protein couple receptor that couples to G proteins of the class GI or GO, and these G proteins, especially the subunits beta gamma, are able to directly bind to KR3 channels and open these channels. When this channels opens, there is potassium efflux, and this potassium efflux will generate IPSPs or inhibitory postsynaptic potentials. Also, there are some evidence some other GPCRs are able to modulate the activity of these channels. Calcium-dependent potassium channels are two major types: the BK or max-decay potassium channels, and these potassium channels require voltage for activation, while the small conductance or SK channels are voltage independent, so they are only gated by calcium. The BK potassium channels are named BK because they have a single channel conductance of 100 picosiemens to 200 picosiemens, while the SK or small k potassium channels have a single channel conductance about 10 picosiemens. Here is a cartoon of a BK channel. What you can see in this cartoon is that the BK channel looks like a KV channel with some additional elements. So you can see the S4 domain, the P region, as in KV channels, but in addition to this, to those transmembrane domains S1 to S6, there's this very large carboxyl terminals or C terminals of these channels. In these C terminals, there is this calcium ball site, which is a calcium binding site. Also, this alpha subunit of these channels associate with beta subunits. So these BK channels are calcium-dependent potassium channels because they activate by calcium, but they are also activated by voltage. They are blocked by if by calcium buffers in the intracellular environment, such as BAPTA or EGTA, or calcium channel blockers such as cadmium or cobalt. Here is an example of single channel traces for some of those BK channels. In C, you can see the dose response relationship between open time in the y-axis and the main potential in the x-axis for these channels. And each one of the curves represents a fixed amount of calcium in the intracellular environment. You can see that varying calcium from 0.1 micromolar on the intracellular side to 100 micromolar, there's a very large change in open probability of these channels. So at minus 50 millivolts, for example, at low levels of calcium in the intracellular environment, these channels tend to stay closed, and that's what's shown in the top part of panel A. Now, if there's 100 micromolar intracellular calcium, now there's a high probability that this channel is going to be open, and therefore in B, at 100 micromolar calcium at minus 50 millivolts, many of these channels are now open. At plus 50 millivolts, on the other hand, these channels are almost maximally open, even if there is 0.1 micromolar or 1 micromolar, or if there's 100 micromolar in the intracellular environment. So these channels are then primarily gated by voltage, but calcium can modulate the gating of these channels. And the action of calcium, intracellular calcium, is to facilitate the opening of these channels. There's many important roles of these calcium activated potassium channels. So for example, it has been well known now that many of these channels evoke what we call after hyperpolarizations. So that's what can be seen here in A, when this after following the action a single action potential of the cell, there's this long hyperpolarization of these cells. Now, if the calcium on the intracellular side is chelated by using BAPTA or EGTA, then this after hyperpolarization is gone, which is a nice demonstration that a calcium activated potassium current originates these after hyperpolarizations. Now, if the cell is depolarized in a sustained fashion, and that's what's shown in B, the buildup of calcium inside of the cells will generate this change in spike frequency that happens with time that we call spike accommodation. So you can see that it is interplay between depolarization, calcium entry, and the existence of this calcium activated potassium channels will modulate the spike frequency and the spike accommodation of neurons.